chapter number 6, Molecular Basis of Inheritance. In this chapter, in the previous session, we have started with a topic called as transcription. We have started regarding the introduction of transcription. So just revise what is a transcription and then we are going ahead. So what is a transcription? Few things you have to remember before I start this topic. So what is a transcription? Transcription means a synthesis of RNA from DNA. This process is called as transcription. Okay, which process is called as a synthesis of RNA from DNA? This process is called as a transcription. Transcription process always proceed in this direction, 5' to a 3'. Which chain of a DNA is going to be copied? Because the DNA has two chains, and in the synthesis of RNA there is only one chain. From this two chain, which chain will be copied? The chain that is going to be copied is template chain. Template chain, that means the chain with a polarity 3' to 5'. This is called as a template chain. And the process occurs in the direction of 5' to 3', means the process of transcription that occurs in the direction of a coding chain. Here the synthesis that also follow the rule of complementarity, means A combines with U, C combines with G, G with C and T with A. There is only one difference here. In RNA the T is absent, so in complementarity A is combined with a U. This is the rule that is going to be followed. The transcription, whole chain doesn't get copied, only a small part gets copied. So there is a transcription unit, and the transcription unit consists of three components. The first one is called as, you can say, a promoter. Second is called as structural gene. And this is third one, is called as terminator. So these are the three components of a transcription unit. Which are the three components of transcription unit? Promoter, structural gene and the terminator. These are the three units of a transcription unit. So let's discuss how this process occurs and what are the enzymes that are going to be required. Okay. So now I am going to start with transcription enzymes and the process that occurs in the bacterial cell and the eukaryotic cell. So let's start with the transcription process. So in the transcription process, the process that occurs in the three steps. The first step that involves in the process, it's called as initiation. Second one is, the second step that is involved, it's called as elongation. And the third step that is involved in it, it's called as termination. The process occurs in the three steps: initiation, elongation and the termination. So this is the three steps of a transcription process. It requires an enzyme. Which enzyme is required? Here the main important enzyme that is going to be required is DNA dependent RNA polymerase. This is the enzyme that is required in the transcription process. So let's discuss how the process occurs. The process of this transcription occurs in a different way in a prokaryotic cell and the eukaryotic cell. The process of transcription in the prokaryotic cell is somewhat simple. So we are going to start with a transcription process in the bacterial cell. So how the transcription process that occurs in the bacterial cell? Okay, so you know that in the structure of DNA, it is a two chain. This is the first chain and this is the second chain which is present. Here this chain is 3' to a 5' chain, and this chain it's called as a 5' to a 3' chain. Because in the structure of DNA, both the chains they are running anti-parallel to each other and twisted around a single axis. The chain with a 3' to 5' polarity, this one, it's called as template chain. As I have already told you that the template chain will be transcribed or copied in the transcription process. Whereas this chain, what we are calling it as coding chain, it will serve as a direction, or that gives a direction in which the transcription process occurs. Means the transcription that occurs in the direction of coding chain from 5' to 3' polarity. Now how the process occurs? Here the first step that occurs is the initiation, the point from where the transcription process starts. So how it is decided? At the certain point or at the certain sequences, the enzyme called as DNA dependent RNA polymerase, it binds. So I am showing you here the enzyme is going to bind. Suppose this is the place where the enzyme is going to bind. Okay, I am showing you this is the template chain. This is the place where the enzyme binds. Along with this enzyme, the enzyme itself doesn't have a capacity to initiate or to start the transcription process. So this is the enzyme. I am just writing the E because I cannot write the whole name over there. So I am just writing this E. This is the enzyme. It binds with a specific site. But this enzyme is not capable of starting or initiating the process of transcription. It requires some kind of an initiation factor. And here the initiation factor that is required is the sigma factor. So the sigma factor is required to start the process called as initiation. So this also combines with a factor, and which factor it is? Sigma factor. So when enzyme and the sigma factor that binds with a specific site in the DNA structure, then the process of transcription that starts or it initiates. Okay, so thus the process of initiation occurs. Now after initiation, the next step occurs is the elongation step. In the elongation step, now there is a role of an enzyme called as DNA dependent RNA polymerase. It brings the complementary base pairs and it will join together. Okay, so here it will join and it form a long sequence like this. This chain is going to be added. There is an addition of a base. Okay, how there is an addition of a base? Like A, G, C, U, A, G. Like this is just for an example. Here these base pairs are added. Base pairs are added which are complementary to a template chain basis. Like suppose template chain per T છે તો સામે A આવશે. If it is a C તો અહીંયા G આવશે. If it is a G then it's a C. If it is a A then U. If it is a T then A, T then A, C then G. So the complementary bases are added, and that complementary bases they are going to be added by enzyme called as DNA dependent RNA polymerase. Okay, and the bases are added and the process continue and the chain is elongating in the direction of 5' to 3'. This step is called as elongation. Now chain is elongating, but at a certain point the termination occurs. The third step that is the termination, where the process of transcription that gets stopped. So it also requires a factor, and here the factor that is required is the rho factor. The rho factor that attaches with it the chain. Rho factor. Okay, this is the rho factor which attaches with it. I am just writing the rho. And due to this, the transcription process ends or terminates. Along with this rho factor, where the rho factor binds, the rho factor binds when there is a palindromic sequences. Before the termination, the palindromic sequences that appear. The palindromic sequences, that means that can be read the same from backward and the forward, they are called as palindromic sequences. Okay, like for an example, if I say A, T, G, C. Okay, clear? So not this A, T, G, G, T, A. So if you read in this direction A, T, G, G, T, A, same A, T, G, G, T, A. These are called as palindromic sequences. This is just for an example. or this is called as a palindromic word. Usually the palindromic sequences that occurs in the opposite chain, but this is just show you where the what is meaning of a palindrome. Like I will give you a simple example. Like see, example rotor. This is a simple word, rotor. If you read in this direction or this direction, they read as the same. Or if I say Malayalam, so this is in backward and the forward direction, it's read as the same. So this is called as a palindromic sequences. So before the termination process occurs, the palindromic sequences occurs, and there is an involvement of a row factor that terminates a process and the transcription ends. When the transcription ends, the chain which is synthesizing, a chain of RNA, this is the chain of RNA which is going to be synthesized, it separates from the DNA. And the DNA molecule which have been unwinded or opened, helical structure which is opened up, that is again winded. So this is what happens in the case of transcription process in bacterial cell or the prokaryotic cell. So how this transcription process that occurs in the bacterial cell? In a 3-step: initiation, elongation, termination. Initiation requires a sigma factor, termination requires a rho factor, and the elongation, that is addition of a nucleoside bases or nucleoside triphosphate with the help of enzyme DNA dependent RNA polymerase. The addition of a basis that occurs in the 5' to a 3' direction, and according to a rule of complementarity, the base are added. So this is process called as transcription. Okay, the termination occurs when there is a rho factor and the involvement of a palindromic sequence. Palindromic sequences means which can be read same from backward and the forward direction. They are the palindromic sequences. The palindromic sequences form a hairpin-like structure, and thus the process will end. So thus the transcription process that occurs in the bacterial cell. But you know the structure of bacterial cell. In the bacterial cell, suppose assume that this is a bacterial cell, there is a presence of a nucleus, but the nuclear membrane is absent. So the cytoplasm and the nucleus, that is not surrounded by or not separated by a boundary, means the process occurs in the same compartment. So there is no more complexity that happens in the case of prokaryotic cell or the bacterial cell. But in the eukaryotic cell, there is a presence of a boundary called as nuclear membrane. So there are two different compartments are there. Okay, in which the process occurred. So what is the difference in the transcription process of a prokaryotic cell and the eukaryotic cell? Let us discuss what is the difference. The usual thing remains the same: initiation, elongation, termination. Even the enzyme remains the same, but there are some more enzymes are also required. The direction remains the same, the coding remains the same, but there are few differences. So just we will discuss what are the differences and what are the different types of the RNA they are going to be produced. So see here, in the transcription I have already told you that it's a synthesis of RNA from DNA. But there are three types of RNA are there. mRNA, second tRNA, and the third one is called as rRNA. What is mRNA is called? mRNA is called as messenger RNA. What it is called? Messenger RNA. tRNA, the tRNA is called transfer RNA. And the rRNA is called as ribosomal RNA. There are three types of RNA: messenger RNA, transfer RNA, and the ribosomal RNA. These are the three types of RNA are there. What is the function of this messenger RNA? The messenger RNA, it is serving as a template. It serves as a template, provides a coding for a next process called as translation. The transfer RNA or the tRNA, it brings or it transfers amino acids based on the sequence that is present on the mRNA. It also reads mRNA base sequences. Okay, so it brings the amino acid based on the sequences that is present on the mRNA, and it reads this mRNA. Whereas the rRNA or the ribosomal RNA, it has structural and catalytic role during a translation process. During the translation process, it has a structural and the catalytic role. So this is the function of rRNA. So in short, there are the three types of RNA: mRNA, tRNA, rRNA. M is the messenger RNA that provide a template for synthesis of protein. tRNA, it's a transfer RNA. It brings the amino acid and read the sequences present on mRNA. rRNA is the ribosomal RNA, that is structural. It plays a structural and the catalytic role. So in the eukaryotic cell, the RNA are produced. All these three types of the RNA are produced, but they are by specific enzymes. This is a difference. So here, first of all, the rRNA is going to be produced by, who will produces this rRNA? rRNA, that is going to be produced by DNA dependent RNA polymerase 1 enzyme. Which enzyme? RNA polymerase 1. It synthesizes all the types of rRNA. There are different sizes of rRNA are there. That 28S, 18S and 5.8S. These all three types of the RNA that is synthesized by an enzyme called as RNA polymerase 1. Okay, clear? Second, the tRNA. This tRNA that is synthesized by an enzyme called as RNA polymerase 2. Sorry, it is RNA polymerase 3. Okay, the RNA polymerase 3, it synthesizes the tRNA. And next, the last one, it's a mRNA. mRNA that is synthesized by RNA polymerase 2. So you have to remember, RNA polymerase 1, it synthesizes the rRNA. RNA polymerase 3, it synthesizes the tRNA. RNA polymerase 2, it synthesizes the mRNA. But the mRNA that is going to be produced, it is not directly synthesized. Here in intermediate, there is one more RNA is going to be synthesized. It's called as hnRNA. hnRNA means heterogeneous nuclear RNA that is going to be synthesized, and then this is processed to a mRNA. Similarly, RNA polymerase 3 synthesizes the tRNA, but it also synthesizes the other type of RNA. Like for an example, it will also synthesizes srRNA and snRNA. snRNA means small nucleolar RNA that is going to be produced by it. So these are the different types of enzymes they are involved. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. They are important. Clear? They are important from examination point of view. So these three different enzymes are going to be required. This is one difference that exists between the transcription process in a prokaryotic cell and the eukaryotic cell. Now the other difference that exists. Now what is the other difference? Here the other difference is that here it's a compartment process. So in the compartment process, the nucleus and the cytoplasm, they are separated, and hence it has to convert into an active form. So what happens during this transcription process? One more complexity that occurs in the eukaryotic cell is that in the eukaryotic cell, you know that there is a presence of monocistronic RNA that we have learned in the previous session. Monocistronic RNA means it consists of exon, then there is a sequence called as intron, then there is an exon, and such kind of a sequences happens. So this is the exon, what I have shown. This is again an exon which I have shown with an orange color, and this is called as an intron. The introns are the intervening sequences which do not required to express, but the exons are required during the next process called as translation. એટલે વચ્ચેનો આટલો પાર્ટ જરૂરી નથી, the intron, and hence it has to be removed. So how it has to be removed? It has to be removed by cutting this. We cut this part. Once we cut, then these are the components which, this is the first exon. For easiness I am showing you this is exon 1, this is exon 2. Now this exon 1 and exon 2, they are joined. And this intron, it is removed because it is not required, it is intervening sequences. So this process that occurs in the eukaryotic cell, that does not occur in a prokaryotic cell, and this process, removing of this intron or the intervening sequences and joining the exon parts in a specific manner or specific sequences, this process is called as splicing. So this splicing, it occurs in the eukaryotic cell, which does not occur in the prokaryotic cell. So this is the difference between the eukaryotic transcription and the prokaryotic transcription. One more difference. Now another difference is that here the process occurs is, you can say that a capping and the tailing occurs. The capping that occurs at the 5' cap means you know that it is applied here at a head. So here the capping that is occurs at the first point, and you know that the process occurs in 5' to 3' direction. So the capping occurs at the 5' end and the tailing that occurs at the 3' end. So what is a capping? Here this capping and the tailing is required to protect the sequence. It doesn't get damaged. To protect it from damage, it is done by capping and the tailing. In the capping, here a specific nucleotide based pair like methyl guanosine triphosphate. Which one? A special one, methyl guanosine triphosphate. It is added at the 5' end. And in the tailing, 200 to 300 molecules of adenylate molecules, they are added at the 3' end. So the capping and tailing is occurred which protect the intermediate sequences. So this is the another thing that happens in the eukaryotic cell, which protect the intermediate sequence. So these are the two changes they are going to be happen in the eukaryotic transcription. First process that occurs is the splicing, and the second one is capping and the tailing that occurs. Second, I have already told you that there are specific enzymes like RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3 which synthesize the respective RNA like mRNA, tRNA and the rRNA. Along with that, the other small quantity of RNA are also produced like srRNA, snRNA, hnRNA. Okay, these are going to be produced. So that's all about the transcription process and the enzymes they are required in the transcription. So that's all about this transcription process. Here we are ending with this transcription. The next process is the translation that we will deal in the next session. We have finished with two processes, replication and the second is transcription. Replication is synthesis of DNA from DNA and transcription means synthesis of RNA from DNA. Now in the transcription, RNA is produced. From this RNA, the protein is produced in the process called as translation that we will discuss in the next session. So till then students, keep learning, keep watching and like and subscribe this channel. Thank you for watching.